Thank you all for coming back, and thank you to our speakers from the last session. We had a great discussion, and I'm excited to continue this discussion here with more biomarker presentations in neurodegenerative diseases that are really relevant here for APBD. So we're going to hear talks from Dr. Neurodegenerative, Associate Professor of Radiology and Radiological Science at Johns Hopkins University, and Dr. David Brenner at the Department of Neurology at Ulm University, and Dr. Orhan Akman, Associate Professor of Neurology at Columbia University Irving Medical Center.
Okay, so first of all, I just want to thank the organizers of this meeting for the opportunity to present some of the new biomarkers that we're developing in our lab. And our great hope is it can actually contribute to this community. So with that, I'll just quickly just move on. So these are my disclosures. And then the outline of my talk today, first, I'm just going to give a very general introduction to MRI. I know there were previous talks on MRI, but as just like a bit of a refresher, I'll talk about how APBD is currently used. How MRI is used in APBD disease, the limitations of these current methods, and then I'll give an overview of a new type of MRI scan that we're developing where we can actually image glycogen. We have just recently started scanning patients with APBD, so I don't have any results to show yet, but I will show some applications to other types of glycogen storage disorders. And then I just definitely want to encourage anyone in the audience, if you have any questions, please put it in the chat and I'll be more than happy to provide additional details afterwards. So magnetic MRI stands for magnetic resonance imaging. It has several advantages. These include this technology is widely available in the community, even at local clinics. It provides excellent soft tissue contrast and relatively high spatial resolution compared to some other neuroimaging techniques. During an MRI exam, a patient usually lies into the MRI scanner, and while they're lying inside the scanner, magnetic fields are turned on and off. And these magnetic fields can cause vibrations in the scanner, and these are the noises that people typically hear. But there's no ionizing radiation. As I said, you're just lying inside a magnet, and these magnetic fields are turned on and off, and it does make some sounds. Typically for these scans, they last somewhere between 20 to 45 minutes, depending on what types of scans you want. One of the very big advantages for MRI is you can actually change the pattern that these magnetic fields are turned on and off, which I'm trying to show down at the bottom here. These are when you turn on the magnetic field and turn off the magnetic field. But depending on the timing of these magnetic fields, the contrast can actually be changed. So within one scanning session, we typically acquire multiple types of MRI scans, and each of these scans give different information, and the contrast changes depending on how these magnetic fields are turned on and off. So there's actually like two types of magnetic resonance scans. The first method I want to highlight is called magnetic resonance, or MR spectroscopy. MR spectroscopy has been around since the 1950s, and it is widely used in chemical laboratories and in biological sciences because it can actually give excellent chemical and metabolic specificity. So it can detect individual molecules within a person's brain or any type of tissue. But one issue is that these chemicals are typically present in very low concentrations. So because we don't have enough sensitivity, we can typically only measure the concentration of these chemicals over a large box. So it's hard to make images of these chemicals. And then if we go to more the conventional types of MRI images that are acquired during a clinical MRI scan, these are actually acquired on water molecules. And water molecules, with these water molecules, we can make a very high resolution image. And the reason for that is water is very abundant in tissue. It has a very high concentration, which then results in higher signal. And this signal can then be used to create maps or images. One of the earlier speakers did mention that he didn't think MRI would be so useful because these changes that they see in conventional MRI scans take years. And he's definitely right. That's one of the problems with the current MRI methods is that these water molecules typically show structural changes. And these structural changes take many years to evolve. One thing that we have tried to do is we try to combine this measuring these actually chemicals, which can give much faster, can actually report much more quickly on changes in the tissue, but we still want the sensitivity that comes with a high concentration of water. So how do we actually combine spectroscopic information about individual molecules such as glycogen, but retain some of that sensitivity from water? So just before I describe that, I just want to reiterate that conventional MRI scans that are used on APBD patients, they typically look at atrophy in specific regions of the brain, so such as medulla and the spinal cord. Also, these lesions are definitely indications that there's some type of demyelination or there's changes in the connections between different brain regions. But as I mentioned earlier, that these changes take a long time to evolve, and they're not specific for what's actually causing these changes. Are these changes just due to atrophy of the tissue? Is the water properties changing? Again, it's very hard to decipher that. If we can actually measure these chemicals directly, it will provide more specific information. So as I mentioned, in addition to APBD, we also developing methods to treat a whole range of glycogen storage disease, or actually assist in the diagnosis, treatment, determining treatment efficacy, and disease load for a whole range of diseases which are affected by glycogen in this case. So I don't need to tell this community how important it is to measure glycogen, but I just want to highlight that in addition to the liver, the concentration of glycogen in skeletal muscle is about an order of magnitude lower, and then it becomes more challenging in the brain because these glycogen concentrations are even lower still, like another order of magnitude. So as I said, what we are trying to do is enhance the signal from glycogen so that even though that the concentration of glycogen, for instance, in brain tissue is low, we can still detect this reliably. Other methods or current methods of measuring glycogen in tissue are listed here. Some of these other methods. Tissue biopsies, as many people know, impractical. It's quite invasive. And also it depends a lot on where you actually basically taking the tissue from. Non-invasive methods obviously are much better, but they have other issues such as low specificity or low resolution. And as I mentioned earlier, these MR spectroscopy methods, they have low sensitivity. So this cartoon kind of shows, probably tries to highlight how can we actually measure a specific molecule, but try to retain the sensitivity of water, like on the right. And the way we do this is that, as I said earlier, we turn on a magnetic field. So this magnetic field is turned on and it's at a frequency which corresponds to the frequency of individual molecules, for instance, like glycogen or creatine. And each of these molecules has a specific frequency which we can tune to. But as we turn the magnetic field on and label these molecules, there is an exchange of this signal from the molecule to water. So if we continually apply a magnetic field to the molecule, This process, it typically repeats itself over the MRI scan, and eventually we build up enough signal into the water pool that we see a reduction in signal. So what we're trying to do is we apply a magnetic pulse at a frequency corresponding to the molecule of interest, but we're observing the signal change on water due to the magnetic coupling between the molecule and water protons. This has several advantages, including that it enhances the sensitivity of our signal. And also a very big advantage is that we don't actually need to inject any contrast agents. This signal can just be turned on and off depending on if we turn on the magnetic field or not. So when we first published our method, we obviously had to do a lot of validation studies. And we did these validation studies in a mouse model. So we had a mouse, and then we looked at the liver of this mouse. Liver is typically present in very high concentration. Sorry, glycogen is typically present in high concentrations in the liver. But after you inject glucagon, this glycogen breaks down and turns into glucose. So we were interested to see, can we actually observe any changes in the glycogen signal after the injection of glucagon? So we performed some baseline scans, and you can see that there's actually a uniform high signal in the liver. But after the injection of glucagon, over the next 2 hours, you can see that there's a gradual decrease in the glycogen concentration throughout the liver. So this was one type of validation we did when we first presented our method about 2 or 3 years ago now. And the next type of validation we did is we did fast these mice for approximately 24 hours. And then we actually measured the glycogen content after we extracted the liver. Again, we want to validate, are we actually measuring glycogen or we're measuring some other molecule? So here you can see that our MRI signal, which is shown on the y-axis or the vertical axis, and our glycogen content, which we measured using a chemical assay, is on the horizontal axis. And there's quite a nice linear relationship between our signal and the glycogen concentration measured using ex vivo chemical analysis. Okay, so as I mentioned, we have just started scanning APBD patients very recently, but I do want to show how the results in some other types of glycogen storage diseases. We actually collaborate with many groups. One is the group of Matt Gentry, Ramon Sun, and others at the University of Florida. And what they wanted us to check is in a Lafora disease mouse model, can we actually see is there an increase in glycogen in the brain of these Lafora disease mice compared to normal mice? And what you can see on the lower left-hand side is yes, the wild type mice shows the heat map is kind of blue, showing like a low glycogen concentration in a wild type mouse. But in these Lafora disease mice, which are approximately 9 months old, there is quite an elevated amount of glycogen, particularly in the brainstem and the cortex. And the distribution of glycogen in these maps corresponds quite well with their immunohistochemistry images. Just if I can quickly just highlight another study we did with Ultragenics in their GSD type 3 or Cori disease mouse model. They wanted to know whether our method is sensitive to treatment effects. So they're developing a therapy, and they gave their therapy to different groups of these GSD type 3 mice at different doses. And you can see that our MRI signal on the left-hand side shows quite a nice dose-dependent amount of contrast across these different groups that we image. And again, there was a linear correlation between our MRI signal on the vertical axis and the amount of glycogen measured ex vivo using a chemical assay. on the horizontal axis. And the maps for representative mice are shown. So you can see in the wild type mice, there's blue, which shows there's low concentration. In a knockout mouse model, it's quite bright, showing there's high glycogen. But when they were given treatment, especially at high doses, that kind of greatly reduced the amount of glycogen. So what I just want to point here is that in this particular study, we were able to detect treatment effects as well. So, and these were imaged 48 hours after treatment. So it doesn't take very long to observe single changes in glycogen in this particular study. And finally, as I mentioned, we have started scanning human patients. This was in a Pompe patients where we imaged their leg. And again, over time, so I just want to say that, yes, we did detect elevated glycogen in the calf and thigh muscle in Pompe disease patients compared to normal controls. So with that, I just want to obviously highlight that we have a new method that we've developed in our lab to image glycogen non-invasively in vivo. So there's no injection of any contrast agents. The patient just lies down inside the MRI scanner and we collect our images. It's sensitive to glycogen levels in mouse models of glycogen storage disease. We can measure treatment effects, and as I mentioned, we have started studies in APBD patients. So with that, I just want to thank our current students and former students in my lab, my collaborators, and also the funding sources.
Yeah, so thank you very much for the invitation to give a talk at the symposium. My name is David Penner. I'm a neurologist in the Department of Neurology in Ulm University in Germany. And my focus is on genetic neurodegenerative diseases, including Lafora disease, but I also have a strong focus in amyotrophic lateral sclerosis and ataxia. I want to talk about wet biomarkers for Lafora disease. And when we talk about wet biomarkers, there are several purposes that they should fulfill. Biomarkers could be useful to indicate a prodromal phase or distinguish pre-symptomatic from symptomatic patients. They should be also diagnostically useful to separate controls from diseased individuals and also from disease mimics, but also could be used for subgrouping or phenotyping patients. They could be useful for to predict disease progression, and also for disease monitoring. For example, also in diseases with relapses and remitting, like multiple sclerosis, but also to see if there's a increase over time or decrease. But they are also, and this is one of the most interesting applications, they could be useful to indicate therapy response, to indicate the clinical response to a therapy. Talking about biomarkers for neurological diseases, obviously it makes sense to focus on proteins that are exclusively expressed in the brain, so brain specific. You all know the well-established biomarkers, tau, A beta, or NSE. But recently, neurofilaments came into focus in a lot of indications. And I want to focus on this biomarker here, and later also talk a bit about GFAP. So what are neurofilaments? It's one of the strongest expressed proteins in neural tissue, and mainly in the axon of neurons, and it gets released once there's neurodegeneration. And you see that it's released to the extra cellular space, and it's a lot more concentrated in CSF than in blood with a ratio of 100 to 1. Or that means it's way easier to measure it in CSF than in blood. But luckily, with the advances in technology, we are now also able to reliably measure it almost as good as in CSF and also in blood. And neurofilaments, there are different components. So it's the heavy, the light, and the intermediate chain. And the light chain, has turned out to be the most reliable biomarker. ALS, amyotrophic lateral sclerosis, is one of the diseases with the highest levels of neurofilaments. We appreciate that it really nicely segregates MND, motor neuron disease, or ALS from mimics, but also from other neurodegenerative diseases like Alzheimer's or Parkinson's disease or facial palsy or polyneuropathy. So it's good really to discriminate between controls and disease mimics. And you also see here on the right side that it's also a prognostic marker. You see that survival is really correlates very well with the levels of neurofilament in CSF. So that's now all CSF. But it's not, of course, it's not specific to ALS because it's a marker that is in neurons, and always when neurons degenerate, this marker gets released, and you see on the right side that it's elevated in a lot of neurological diseases, but the most it's actually in ALS and also in Creutzfeldt-Jakob's disease. So it's not specific to ALS, of course. And as I mentioned, we are now able to measure it in blood also very well with the new technologies, with ELA and Simoa. And this is, I think, is a decisive change because blood is much better available and also longitudinally available. So you cannot take CSF at every visit, but you can take blood at every visit. And you appreciate here that it discriminates ALS as well as in CSF. And it's also very good to discriminate from disease mimics. So that's why it's almost routinely used in ALS diagnostics now, and precise cutoffs have been defined. And you see here also again its suitability as a prognostic marker. So in ALS we have different phenotypes, so not every patient has the same disease progression, and it really correlates well, and you can predict disease progression according to the level of this marker. One problem is that the neurofilaments can be influenced by a lot of factors, and the main factor is actually age, what you see here in this graph. So age, renal function, but also obesity, diabetes are the main factors that influence the levels, and that's why in cohorts it's with normal controls, they have been measured, and precise age-dependent reference values have been defined. So you always have to interpret your neurofilament level against these reference values. And well, now I come to what I mentioned, that I think the holy grail of a biomarker is that it can predict or indicate a treatment response. And in ALS, we recently got the first specific therapy for one genetic ALS form. So ALS caused by mutations in the SOD1 mutation. And these mutations have a gain of function, and that's why ASOs have been developed against SOD1 RNA. And this therapy is really successful. and you see that upon therapy compared to baseline, there's really an impressive drop that correlates with the improvement in the functional scale in this disease. And you also see it on the individual level. So not even need the group level. You see it on the individual level that this therapy is working. And that's what makes it neurofilaments in blood a really good biomarker. I will, of course, later talk about neurofilaments in Lafora disease, but before I want to also talk a bit about GFAP. So it's obviously a astroglial marker, and we know that it's actually a good marker of astrogliosis from a genetic disease caused by gain of function mutations in the GFAP gene. So that's why it has also been studied in other diseases where we know that there is astrocytosis, like in multiple sclerosis. This is now a measurement in CSF, and you can see that there is a difference between controls and different forms of MS. So it depends on the form, where you see the best discrimination. But meanwhile, it also been established in serum, and you see also that it has a value in MS, and it's even more elevated when MS patients have a acute inflammatory lesion. I think what is one of the most interesting applications is defining it or combining it with neurofilaments. You see it here. So here's the proportion of people with disease worsening, and this line here reflects the group with the greatest worsening, and you see that this group is characterized by the highest levels of both GFAP and neurofilaments. So really interesting finding. But now I will come to the actual topic of my talk is about neurofilaments and GFAP in Lafora disease. I have to say we have not measured GFAP yet. That's why I want to focus on neurofilaments. And so why is neurofilaments and GFAP, why are they good marker candidates in Lafora disease? So we know neurofilaments, obviously, as I told you, has been established as a really good diagnostic, prognostic, and therapeutic marker in ALS. So we fundamentally know that it's really a suitable marker, and we know that there's neurodegeneration in Lafora disease, also including axons, of course, and we know that there's astrogliosis and in Lafora disease, and from mouse models we know that astrocytes really play a decisive role in disease modification in this indication. That's why we decided to measure both markers, but as I said, we started with neurofilaments in a cohort of German Lafora disease patients, so 14. So this is our cohort in detail. Actually, we have in bold is indicated one mutation that is known for especially mild disease progression. So we actually have a lot in Germany. And the basic characteristics of these cohorts are shown here. So we have patients in all disease stages indicated by the Lafora disease disability score, and while our group was similar HDF onset as other published publications. So we have 16 patients that are still alive and 3 that already have disease. And when you now look, so as I said, we measured serum neurofilaments in this cohort, and I plotted it against the age in this graph. So it doesn't look so impressive. You see in the lines you see the percentiles of the normal values, the age dependent normal values, and you see that some patients do have pathological values above these reference values, but most are below. But so if you plot it against the duration of disease, it becomes a bit clearer that there's a correlation, so that levels are higher the longer the disease runs. But you see that there's a group that does not fit in here, and this group is this mild mutation. But when you plotted the neurofilaments against the functional scale, then you see that these patients, these mild patients, also nicely correlate with neurofilament levels. Yeah, so neurofilament levels correlate better with the functional performance scale than with disease duration. And when you look at the subdomains of this performance scale, you see that there is a correlation for every subdomain. So the worse the patient is on the subdomains, the higher are the neurofilament levels. And when you make a bar graph grouping according to the disability score, so we define 4 scores. So 4 is the worst, and score 1 are mildly affected patients. You see that it increases with increasing disease progression, and maybe that may especially be a marker for the late disease stage. Of course, one would wish that you already see more dynamics in earlier disease stages, but on the other hand, thinking about a therapy also for progressed patients and wanting to see a therapy effect that it could be still useful in these progressed patients if you maybe see a decrease in neurofilaments with a working therapy. So the thing is that you see is obviously that we still have a low number, and we have to confirm this data in a larger population. And that's why we teamed up with the centers of Bologna and Dallas to make a much bigger cohort, and we are shortly before measuring them all together. So in the end, we will have a cohort about 40 patients, and I think this will bring clarity if neurofilaments are a good biomarker. But as I told, so we will not only measure neurofilaments, but also GFAP, because it's obviously also a really interesting candidate. And with that.
All right, thank you. Thanks again for inviting me for this interesting topic. We have seen many approaches today, and all of them are very creative, and they actually gives physicians option to follow the patients and the progress of the disease without spending too much money or taking the patient's time or tissue samples. And now I'm going to mention the branching enzyme activity as a biomarker, which is the main reason for APBD that actually caused the disease, decrease in the branching enzyme activity. And so I just going to mention a little bit about branching enzyme again. So branching enzyme branches glycogen, and it is the mechanism is relatively simple. It doesn't require any cofactors or anything except the long chain of glucose, and it just transfers the glucose units at the end where the glycogen synthase adds and makes an alpha 1,4,1,6 branching point and makes a branch for glycogen synthase to continue again to synthesize the glycogen. And if the building block is glucose, but in terms of glycogen synthase, it is UDP glucose, which has been to favor the reaction to go forward. So this of course, in the apps, our purpose both for treating the patients or understanding the disease to manipulate the GB activity most of the time to, of course, to increase it. And to be able to test it, we use the method that is quite different than the synthesis. So this shows the polyglucosan when there is no branching enzyme activity or less activity versus the normal glycogen molecule here. And to be able to determine the enzymatic activity in vitro, we use different enzyme than phosphorylase. In the tissue samples or the cell extract that we have, we use phosphorylase A with the glucose 1-phosphate. And in this case, we, instead of using glycogen synthase, we reverse activate the phosphorylase and add the radiolabeled glucose into the growing chain by phosphorylase activity. And of course, the sample that we collect from patient or the cells where the branching enzyme expected to be cleaves the glucose unit and then makes the branch here, just like in the cell. And phosphorylase, just like glycogen synthase, continues to synthesize the glycogen from this point onward. So if everything is okay and if we have the normal branching enzyme activity, this reaction generates a glycogen molecule that is highly radioactive because of the radioactive glucose 1-phosphate we put into the system. And if there is any defect, then of course phosphorylase continue to synthesize the chain. It adds some radioactive glucose, but this is very minimal. So it is most of the time logarithmic scale, and in most of the case in our samples, we don't detect any activity. However, I have to mention here that the tissue sample that we choose, I have choose personally from patients or from the other animals, instead of muscle or liver, where the branching enzyme activity was supposed to be the highest, I prefer to use white blood cells where there is not much glycogen at all. And the reason is because of that, phosphorylase or glycogen synthase can continue to synthesize and add the radioactive glucose, and this generates a false positive, or false negative, I should say, because you still see the increased amount of activity. Of course, this is only related to amount of polysaccharide in the tissue. White blood cells has almost none, very few, and therefore they generate very low background, and then we are able to determine the enzyme activity, I should say, perfectly related to the mutation or amount of the GB in the tissue. And so using this method, actually, from the white blood cells, we are able to detect the glycogen branching enzyme down to even less than 10%. So we were able to detect that the homozygous patients for PY329 mutation had like 20% enzyme activity versus compound heterozygous patients, which they totally lose the other allele. We were able to detect like 6 to 7% enzyme activity, which actually very well relates to the genetic condition. And that's why I think this is the best method for determining any kind of treatment that targets the enzymatic activity. And these treatments are basically the gene therapy that directly increase the branching enzyme activity in the tissue because of the new gene that's given to the cell, or antisense oligonucleotide therapy that targets the correcting the mutation in intron 15 in GB1 gene. In those treatments, of course, increase the enzyme activity, and we are able to detect this by using GB enzymatic activity. Yesterday and some today, we have seen other treatment options that may decrease the glycogen synthase activity or to target the polyglucosans get degraded using the ORS compound. And in that case, branching enzyme activity is not being impacted, increased or decreased. So in that case, this enzyme assay is not a good indication of the disease condition. And but I also have to mention that in gene therapy or antisense oligonucleotide treatment, the treatment may be given directly to the brain through intrathecal injections. In that case, it is of course only the brain tissue is going to have the treatment, and enzyme activity may increase in the brain tissue, but it will not impact the other organs like muscle or the blood in the patient. In that case, of course, we cannot use this. But of course, we will still try to determine the enzymatic activity in CSF cells, if there are any. This method is very sensitive. If we have like few hundred cells, we can detect the enzymatic activity very well. And so this is will remain to seen if we are going to be able to get enough samples from the patients or from CSF that we can detect. I know that Dr. Nitschke tested CSF for polyglucosan, and that was it is promising, yet enzyme activity may not be detected in the CSF. So now this is an option. Of course, enzymatic activity is not easy to make. It's not clinically available. No commercial company is doing this. If you know, please let me know, because we contacted by several companies. They didn't want to continue on adopting the methodology. So, but we can do it in the lab, in the research basis, and I think this is the good way of at least measuring if the treatment affects the enzymatic activity. All right, so that is all that I'm going to present in terms of branching enzyme activity. So if you have questions.
Thank you so much for these three great presentations. We really appreciate that. We do have some time for questions. We're going to go to 1:40 for this session. And so we'll have a 5-minute break afterwards. So please, you feel free to keep adding questions. So I'm going to go with this first question for Dr. Brenner. Since clinical labs such as LabCorp in the US use a proprietary method for testing NFL and other metabolites, while Simoa uses a different platform, yet both use the same Quanterix antibody, how do we tackle comparing the results between clinical NFL reports versus research NFL reports when there isn't a conversion factor available by the clinical labs?
Yeah, so this is always a problem. I think it's important that you establish it in-house, and it's very good validated with control so that they give always the same values. So, but this is internally. I think what will be the future is to use set values. I think, and then you can also pool data a lot better. But so set values reference to the internal normal values, and that can be used in international cohorts. Yeah.
And I think establishing the normative values across the assays will be perhaps helpful. But I think one of the things we don't know is, particularly for APBD, what the values are that we should be worried about.
Yeah, so this would have to be defined. If they are elevated at all, you see that in Lafora disease they are elevated at a later disease stage, but not at the early disease stage. How it is in a polyneuropathy, could be higher because yeah, but this would have to be measured. Yeah, for example, in Guillain-Barré syndrome, we also find high values, or in vasculitic polyneuropathies, they're also high. Yeah? So this would need to be checked. And as I said, it's really crucial to normalize them to age and also probably renal function.
There was actually another question about age. How do you add that in for someone with APBD with late onset to take out the age factor to know it's truly elevated?
So well, we always plot it against the percentile lines. So if we do it on an individual level, and if you get get your result, you see your data point in relation to the normal values or the percentiles, and this you can also recalculate to a set value, and with this we can make very good calculations. So the value is not dependent, or you have out calculated the age factor.
Thank you. So it's certainly possible. Dr. Yadav, I have a question about your approach. Is this done on clinical standard MRI or research grade like 7 Tesla MRI?
No, so definitely our lab, one of our primary motivations is to translate any methods we develop into other clinics. So we've been focusing our efforts on 3T standard MRI scanners. There's no special hardware required. We do make some software changes, and then I'm currently working with several groups to try and translate the methods we're developing here and implementing them at other sites as well. That's ongoing at the moment, and yes, definitely, it's 7T scanners will give you a more specific image, give you like a higher resolution, but we're focusing on trying to implement this at 3T just in terms of translation.
Great. Okay, so I have a question for Dr. Achman. How do the variations in white blood cell counts in patients and controls impact GBE1 activity assays?
Actually, it doesn't affect at all. So the enzymatic activity is directly related to the amount of protein that we extract from the cell. And it is, if there's a low blood count, it doesn't matter, we always correct it to the protein. And the correction pretty much gives the same value. And so either in the blood or I have to mention that any tissue that we measure in the body, the enzymatic activity is pretty similar from tissue to tissue. Even muscle or liver that's supposed to have higher activity, which they do, but this is due to the glycogen content. And actually, at least the enzyme assay that we have significantly affected by the glycogen amount in the tissue. So it gives higher activity. But if we have chance to get rid of the glycogen and start from a scratch with the available enzyme, then we will probably get the same enzymatic activity for blood cells versus any other tissue. And well, having said that, we try to get rid of all the glycogen in the tissue, like muscle or liver, and measure it again. So branching enzyme strictly depends on the existence of glycogen. If you remove the glycogen, the branching enzyme kind of falls apart and you don't detect any activity. So it is one of the limitations that is while isolating the branching enzyme or measuring its activity. So it is to have some inherent initial glycogen content.
So one question for all of you, you know, the glyco-NOE could be specific for APBD. GBE1 is certainly specific for APBD, NFL, and GFAP are not. It's used in many other disorders. But I think Dr. Brenner brought up the idea of a composite and comparing it with other things. And how can we think about our studies going forward to combine multiple factors to see if, you know, in clinical trials, we need to combine these factors as our primary endpoint? Or is that the goal, or just one at a time? Just curious.
Yeah, maybe I can go first. So in terms of the MRI scan, especially with new methods, you definitely need some type of validation. So I saw the question earlier about age-matched control. So that's like definitely a first step. Just like comparing the results we're getting. I can talk specific about the MRI method and comparing that with age-matched controls. And the second one is also comparing the results across sites. So in terms of MRI technology, especially with new methods, there's often quite a variation depending on where you get MRI scan done. So just increasing the power of the measure that we're providing, I think that can be improved by firstly translating methods to other sites, writing down a protocol where every site is performing the scans in the same fashion, and then seeing are the results reproducible or not. So that doesn't exactly address the question about combining different measures, but yeah, hopefully that gives additional clarification.
Absolutely.
May I just say something to Dr. Yadav? And if the MRI strictly depends on the amount of glycogen, I mean, the branching enzyme deficient mice, YS actually has a very high amount of polyglucosan in the tissue. So I think I am not sure about the patients. I never had a chance to examine a autopsy sample, but it will definitely give a very impressive result.
Yeah, so one thing that I also want to kind of add is I also working with Felix and my young Dr. Varma. So we're hoping to kind of validate our MRI measures with the different types of readouts they're getting. So I think hopefully in the coming like short amount of time, we can get these additional measures and again, just like validate or increase the power of what we're measuring.
Maybe I can add a biomarker as a primary endpoint. I think we're still far away, and maybe it's also not a good idea because in the end, it's a marker, and what we want to know if the patient clinically benefits. I think it can be really useful to reduce patient numbers to see if there's a signal, and then make a go or no go decision if you move forward with a substance to a clinical endpoint based study. I think this is but this would already be a really good. And I think this is how we now in ALS, where it showed you the highest levels and where it really drops upon therapy. So in this the indication, we now are on the point of doing biomarker driven pilot trials, and then decide if we go if we do a controlled disease marker or clinical marker based trial.